I would like to introduce our first speaker. This is Maciej Mrugala. He is a board-certified neuro-oncologist. He graduated from the Medical University of Warsaw and then completed his clinical residency at the Massachusetts Medical School. Now he works as a professor of medicine and neurology at the Mayo Clinic in Phoenix, Arizona. The micro is yours.
Thank you. Thank you so much. Thank you for staying. I know it's been a super long day. The subject I'm going to discuss, neuro-oncology, is not the most uplifting subject for Saturday afternoon, but I will make sure that I will share with you some really promising developments in this field, and we are all very excited about that. Of course, thank you to the organizers for having me. It's a great pleasure to be back in Boston. I actually trained just across the river at MGH. I lived in Cambridge by Harvard Square and took Red Line every day to work, so it really feels like home. So my job for the next 20 minutes would be to share with you some of the developments in our field, and I don't really have a disclosures. So glioblastoma, many of you know, it's the most deadly form of human cancer. It is unfortunately increasing in incidence, and the survival rate continues to be dismal despite many clinical trials and studies, with 90% of patients recurring very quickly, and most of them, unfortunately, not living more than just 1 year. So in the old days, we have been using the tools that were helpful, but not completely helpful. And one of these tools, as you can see on the right, is an MRI showing the enemy, as I call it, and identifying that the patient has a problem. And on the right, you have histology, so typical H&E staining. That's how we would diagnose brain tumors, several years ago, a decade ago. Kind of looking at what it looks under the microscope. There are certain features like vascular proliferation and necrosis, and that would kind of lead us to the diagnosis. However, things have changed quite dramatically over the last couple of decades, and then we have been witnessing a really molecular revolution in the field of neuro-oncology. This slide I borrowed from my colleague, Craig Horpinski from Northwestern, and what you have here are the 4 editions of the WHO classification for brain tumors. The first one is from 2000. It's actually funny because it sort of follows my career. So 2000, I was a resident in Worcester at UMass. And 2007, I was at MGH. 2016, I was at Fred Hutch and UW in Seattle, and now I'm in Arizona. So you can see I was kind of following the development of the molecular pathology in neuro-oncology, but you can see also that it basically almost doubled in the size of the book, which is good news for us in terms of understanding the biology of this disease, but also plan the treatments. To paraphrase another Nobel laureate, Henryk Sienkiewicz, Quo Vadis, everybody knows this novel. So neuro-oncology, where are you going? And is there really one glioblastoma or maybe more than one? And as I mentioned, this genetic and molecular revolution led to development and basically discovery of genes that are associated with a formation of these tumors that allow us to classify them, and they also allow us to pick treatments for these patients that in the end can make a difference. I show this slide frequently as it is somewhat depressing. This is the FDA-approved list of therapies for this tumor, glioblastoma. As you can see, it's very short. And the last approval was actually 10 years ago. So for 10 years, we didn't really have a lot of options for this disease. But thankfully, the landscape is changing. Just a couple of brief slides on how this tumor is being treated in the past. Most of you know that after surgery, patients would get radiation and chemotherapy. And this study from 20 years ago showed for the first time that addition of chemotherapy to radiation makes a difference. And this study showed that when you do this, the survival improves by about 2.5 months. So you can laugh at this, right? 2.5 months, what is it? But I mentioned to you at the beginning that the survival in this disease is about 12 months. So adding 2.5 months to somebody's life was meaningful, and this was, as you can see, statistically significant. And then after this study was done, we started to understand the molecular makeup of this tumor even better. And we found that some patients who were on this study lived twice as long as the patients that they didn't have a specific mutation. And this kind of started really the process of us being very selective in terms of who should get what treatment and how this treatment can affect the patient's life. So the era of same for all has ended, and we are now really much more selective in terms of how we treat patients with this disease. One of the areas that I was working with were tumor treating fields. As you know, electrical fields have multiple biological properties depending on the frequency and intensity of the electrical field. You can alter the biology of the tissue, you can destroy the tissue, you can also modify how the tissue works. And tumor treating fields of specific frequency of around 150 to 200 kilohertz have the ability to stop the mitotic process. So basically, it's an anti-mitotic treatment. If you remember, there's a lot of scientists here, so mitosis is not foreign to you. So tumor treating fields are typically affecting different stages of the mitotic process. And similarly, some drugs do the same. But tumor treating fields seem to be kind of working more along the cycle of the mitosis. So this was translated into the clinic, and device was built to be worn by the patients. And you can see it better a little bit here. So patient diagnosed with a brain tumor, then we do the treatment mapping. So you want to make sure that the treatment arrays that are here are applied to the scalp in such a way that the intensity of the field is the highest where the tumor is. And then you can measure the compliance, meaning for how long is the patient actually using the tumor along the 24-hour period. And it was found that compliance is associated with better survival. And this treatment was approved by FDA based on this clinical trial that showed few things. First of all, it showed that many patients, 43% of patients, were alive at 2 years. So remember the first slide, 15 months, right? So this is better. This is better than before. And also, I want you to see, look at the kind of the tail of the curve. 5-year survival in this disease is unheard of. But with this treatment modality, about 13% of patients were alive at 5 years. So this moved the dial, not as much as we would have wanted, but nevertheless moved the dial. And about 10 years ago, this treatment was approved for treatment of glioblastoma by FDA. And this was the last treatment that was approved for this disease since that time. We are awaiting something to come up pretty soon. So when this was approved, my group took on a kind of a follow-up study. So as you know, in research, when you do a phase 3 study and then the drug is approved or a treatment is approved, then it's launched and it's basically used in the clinical setting. And then you can do what is called a phase 4 study. So you can kind of follow how this treatment is actually doing in clinic and how is it affecting patients. Is it working the same? Is it working differently? Are there any new side effects? So we did this for patients with recurrent disease, over 400 patients, and we kind of confirmed the results from the clinical trials, and also had some interesting findings, kind of showing, for example, that in some cases the patients were actually doing better than they were on the clinical trial, which I'm not going to go deep into the explanation why, but again, importance of phase 4 experiences. And then last year we looked at 25,000 patients, so it's really a very large cohort, patients who have received this treatment in the clinical setting, and we kind of confirmed what was found in the earlier studies, and specifically in terms of safety. Never forget about safety. When the drugs are launched based on, let's say, 500 or 600 patients, and then there's thousands of exposures, so we can gain so much more data from those actual exposures, and we frequently forget how important phase 4 studies are. So this modality, in addition to being anti-mitotic, also was found to have ability to affect migration and invasion of the cells. There are some signals in terms of changing immunological milieu of the brain, altering blood-brain barrier and others. And as you can see, It's now approved for glioblastoma, as I mentioned, mesothelioma, very rare form of lung chest cancer, and metastatic non-small cell lung cancer. And there's ongoing research also in other diseases. So I think you will see more and more of this technology, which we call a wearable treatment, in the very near future. So let's just summarize quickly the landscape for this part of the talk. So surgery, radiation, then you do adjuvant treatment, but then what you do next at the time of recurrence. And this is where the immunotherapy revolution comes to play. Many of you, I'm sure, have heard about checkpoint inhibitors. These are drugs that are eliminating the overactivity of the immune system and are basically unleashing the immune system to fight cancer. You've heard about chimeric antigen receptor T cell therapy, which is basically a genetically modified T cell process. And of course, viruses have been used for a while. So immune checkpoints, just to not spend too much time on this slide, is just basically removing the brakes from the immune system and allowing the T cells to find cancer cells and kill them. There was a Nobel Prize for this in 2018. And this approach has been tried very successfully in melanoma, with many patients surviving melanoma for years now, and in other cancers as well. Not so much in brain cancer as of yet. Some of the studies I'm showing here were negative, but we continue to study this process. The second treatment I wanted to bring up today are CAR T cells. So these are engineered modified T cells with chimeric antigen receptors that can be very specifically programmed to recognize specific cells and attack them and then destroy them. So this is a very new concept of basically living cells treating the cancer. So then you can see them here. You can design them and engineer them based on the molecular signature of the tumor. And in gliomas, we specifically usually use EGFR, IL-13, or CD19. So these are some of the treatment targets. The process is relatively simple. It is maybe analogous to stem cell transplantation. You basically collect the cells from the patient, then you expand them in culture, then you do the gene transfer, and then you expand them and again put them back to the patient. So it's really pretty straightforward. Of course, it took a while to develop it, but now it's a very easy process to administer in clinic. In a few studies I wanted to share with you with this approach. This is from UPenn group, looking at the bivalent CAR T cells. And just one thing to remember from this slide, so a few patients that received this, you can see just right after the infusion of CAR T cells, the white area is the tumor. You can see how quickly it disappears from the tumor, from the scan, which is extremely uncommon in gliomas. We don't really see this. Small study, nevertheless quite successful. Another study that is worth sharing is one of the first studies in this space done at the City of Hope with IL-13. I was privileged to be actually taking care of this patient who was a physician, and he was the first one in the world actually who received CAR T cells intrathecally. What it would mean intrathecally, directly into the spinal fluid. So he was treated both with the cell injection into the brain and into the spinal fluid, and you can see here that his tumors have basically melted away. And he did well for a while. The main issue with this approach, even though it works so beautifully, is the durability. So after a while, unfortunately, most patients would have progressed. And we're kind of working on how to solve this. So how to overcome this? Combination approaches, next generation CARs, and some advanced delivery methods. Few things about novel delivery systems that we've been working on, specifically using the blood-brain barrier disruption. As you know, blood-brain barrier is frequently a main obstacle for chemotherapeutics to get into the brain at concentrations high enough to be cytotoxic. So there's some work is being done in this space. At our institution, we're currently using a device that is implanted to the patient. And the patient is then treated with a chemotherapy drug, in this particular case carboplatin. At the same time, he is receiving a sonication. So basically, you can see on the right side of the graph a sonication needle that is placed, this little disc that you see is right here under the skin. and we place this needle, sonication needle, in this space. We sonicate for about 5 minutes while the patient is getting chemotherapy, with the hopes that the drug will penetrate the blood-brain barrier and get to the brain tissue very, very easily. As a matter of fact, this patient was treated yesterday, right before I left for the airport, and he's doing well, so I'm really happy to report. And then lastly, something that I wanted to share with you in the space of low-grade gliomas, IDH inhibitors. So what is IDH? So IDH mutations are very common in patients specifically with low-grade glioma. So glioblastoma is a high-grade glioma. In the low-grade glioma space, we have oligodendrogliomas and astrocytomas, and many of them have this mutation. And the mutant IDH enzymes produce D2-hydroxyglutarate, which is an oncometabolite, and it promotes the growth of the tumor. So there was a lot of work being done to see if blocking this process could result in a positive outcome, and basically using a concept of the druggable target. And this was successfully done. The study using the IDH inhibitor, vorasidenib, was published in 2023, showing a significant improvement, as you can see on the Kaplan-Meier curves, in the survival of patients with lower-grade gliomas, counted in months and years. And this drug has now been approved by FDA, actually August of last year. So we have a new drug for low-grade gliomas with IDH mutation now available to our patients. And then just the last couple of slides. Dr. Nowakowski previously said in his talk that we should be using every possible tissue that we have and not waste any material from our patients, and I couldn't agree more. And one of these tissue specimens that we frequently waste is CSF. And CSF can be a window into the nervous system and can really be something that we can use and call it as a liquid biopsy. So basically, what can we look for in the CSF? We can look for circulating tumor cells. We can look for circulating tumor DNA and microRNA. And all of this can give us valuable information. There are differences between these three technologies, but nevertheless, they are all basically liquid biopsies. And I wanted to share some examples from my practice showing a patient with breast cancer who had negative conventional cytology. So the way to find cancer cells in CSF is typically to use cytology, and cytology is very low. Has very low sensitivity. So using this methodology, we were able to identify the DNA in a patient's CSF and start the treatment. This is another case of a person with ovarian cancer, also detection of cancer cells with this methodology, then treatment, a drop to zero, then stopping treatment, escalation, and then treatment again. So you can not only use this technology to diagnose the condition, but also to monitor the patient along the way, which I think is very, very valuable. So in terms of applications, I think that we now know that this methodology can be very helpful in the detection of cancer to begin with, even if there is no other ways to see it, specifically on imaging. You can do molecular profiling, so not only that you can find the presence of the cancer, but you can then do profiling to identify which genes are driving this and are they potentially druggable, so are the targets. You can also follow residual disease, and you can do like serial analysis. So basically, it's like monitoring sugar, blood sugar in somebody with diabetes, right? You have a disease, you have somebody with cancer, you treat them, they have positive liquid biopsy, then you convert to negative, and then you follow them longitudinally, testing along the way, making sure the disease is not coming back. So this is actually already happening in practice, and this is a huge development. And one other comment that sometimes liquid biopsy can alter the treatment. This is an example of a patient with an astrocytoma who was treated previously with chemotherapy and radiation and then had a progression. And at the time we did a liquid biopsy, and what we found was a specific mutation called 1p19q deletion, which typically indicates sensitivity to a specific drug called temozolomide. And based on this finding from his CSF, we altered the treatment, which then allowed him to live for almost 2 years after the diagnosis. So you can see that this technologies can be life-altering for patients and can really allow us to be very targeted. This is just his sample of the genome. So just to summarize, I think we are observing a very rapid development of diagnostic and therapeutic modalities in neuro-oncology, which is very hopeful. Liquid biopsies are already being done in oncology, mostly on blood, but CSF is the space that we can use it on very easily. We now have the first IDH inhibitor available and in clinic, and there will be others coming up soon. Watch out for the CAR T cells. They are marching into solid tumor space. They're already very successful in leukemias and lymphomas, but I think we're going to be using it in gliomas very soon. And then I showed you that some of the device-based treatment therapies, so either tumor treating fields or maybe sonication can be helpful. And just to finish off, because we are at the Polonium Foundation meeting, I just wanted to share that we just formed a Polish Society of Neuro-Oncology. This happened last year in Bydgoszcz, and the inaugural meeting of the society will be in January 8 and 9 in Bydgoszcz. So something to be proud of, that Poland is going to be on the neuro-oncology map of the world, and I would like to invite everybody who is interested to come to Bydgoszcz in January. And thanks again for your attention.